Yes, yeah. This is going to be the 12th session, and this is going to be the last session. We had a session yesterday. I think the NPTEL team did send the mail about this, about there's going to be a session yesterday, but I don't think many people saw it, or many people were not able to join, I suppose. I'll also like first finish the 12th session, and then maybe go back and give a little bit information about what was there in the 11th session as well. So I'll start sharing the screen, and I'll start with this session. All right. Yeah, is the screen visible in full screen mode?
Right.
Yeah, okay, awesome. So yeah, today's session is this is the last session, like I mentioned. This is going to be more about applications. So far we saw a lot of techniques. In between we did see some examples and applications, but this session is completely dedicated to looking at real-time applications and case studies, and what is the current problem. uh, that is what will be addressed in this session. So emerging pollutants is just to get an overall idea about what are all the problems that we need to deal with. Now that we have looked at various techniques and how to solve issues, it is important to take a step back and look at overall problems that are currently there, and newer problems that are being created, so that we'll be able to resolve them effectively. This chart gives us an idea about emerging pollutants. So pollutants like carbon dioxide, greenhouse gases, and chromium, and so many of these metal compounds. These things we have yeah, looked at them over and over. We know that they are pollutants. We know they are causing problems, but there are also problems that are newer problems that are emerging right now. They are not of a big issue, but they have the potential to become a bigger issue, and most of them are biotechnological products, or newer developments that did not exist like probably a decade ago, and that can be attributed to hormones and pharmaceutical products, human products, human waste, these things that are increasing, especially with viruses like COVID that is coming up now. These things did not exist in the past. Now this COVID associated waste also adds up to this list of emerging pollutants. So this gives us an idea about what all could be this emerging pollutants. um, Someone has any question or something? Okay. Yeah. Even a lot of cosmetic products and personal care products, which were not the way it was a couple of years ago. Now with a lot of innovations and biotechnological applications happening, these are also becoming waste products that need to be dealt with. So this list is an overall gives an overall idea about what is the source or the origin of these emerging pollutants, how they are affecting human health and environment, how long can they stay in the environment, and how much they can get accumulated in the environment, and what are all the different components of the environment that is getting affected because of these chemicals. For instance, you can see steroids and hormones, veterinary medicines, phthalates, bisphenol compounds. And it's very surprising to know that many of these are coming from human use products, or products that humans have started using to make their life better, which did not exist a couple of decades ago. And as you can see in this, we don't find the common pollutants that we always look for. They are not in this list. These are all newer components that are emerging. So how do we control this emerging pollutants? I'm sorry that this image is kind of pixelated, but yeah, so the common ways that it can be done is adsorption, membrane technology, oxidation, constructed wastelands. So constructed wastelands has become one of the major ways for controlling many of these emerging pollutants, like medical waste, clinical waste, and even nuclear waste is being controlled with this particular method, where um, um, like we create places where we can dig up and dump all of this and then close it, and build construction over that, and it'll and assume that it will forever remain there. But the problem is, there's always going to be leakage. There's always, if something happens and the controlled system damages, it's going to seep in to soil, and it's going to get into the groundwater and contaminate water bodies, and get into the system through plants and so on. So this is not exactly the best way we would want to control it, even though constructed wetlands have become a really popular way of controlling these emerging pollutants. But it can also be controlled by other ways, through synthetic pathways. So here there are various examples of synthetic formic acid and carbon dioxide fixation pathways. So these carbon-based compounds, and the carbon dioxide release can also be fixed, not by so we know that carbon dioxide can be naturally fixed by plants, and it can be fixed by grassland ecosystem and so on. But it can also be fixed through these certain synthetic pathways, which can be induced in microorganisms. So since we've been reading about microorganisms, we can induce these synthetic pathways in microorganisms by modifying their genes. Genetic engineering can help us in doing this by introducing a gene which will now help them in fixing carbon dioxide through either of these pathways, and help us in controlling some of the emerging pollutants. Carbon dioxide can also be fixed. So we are shifting more towards carbon dioxide fixation now, and seeing how microorganisms or other greener ways of doing this. So carbon dioxide fixation can also be done by microalgae, which can use this carbon dioxide and convert it um, um, into biodiesel. So microalgae can be employed to convert flue gas or syngas to biofuel. So syngas is also another emerging pollutant. Syngas means synthetic gas. It's like a gas product which is released from industries, which contains all sorts of biohazardous chemicals in the form of vapors, such as volatile organic compounds, and that gas is what is termed as synthetic or flue gas. So natural gas, we might have heard, is like methane and all, which comes from natural degradation processes, natural biodegradation processes, um, um, or decomposition processes. Those are natural gases, and we have ways of using natural gas for our own purposes. But this flue gas or synthetic gas is often released into the environment and mixes and becomes greenhouse gas, and causes harm to people, causes harm to the environment, causes harm to animals, plants, and everything in the environment. So flue gas is a major issue, and releasing cleaner gas is something all the industries should take up and look at. And microalgae is one of the solutions that many industries have now employed to convert flue gas into biofuel. So microalgae can take up these different heavy metal and sulfates and nitrates, and use that for its growth and produce biomass, and also produce energy. So microalgal biomass is produced, and carbon dioxide is reutilized and converted to different pathways to produce biofuels, etc. So this biofuel can later on be used for various other purposes, and can be repurposed in the industry itself for some other processes. So this is how a microalgae works. Yeah, algae are, again, they have photosynthesis. They are autotrophs, so they take the energy directly from the sun's light. They only need the electron acceptor and donors and other nutrients, because they are using the light energy as the energy source. So we don't have to, if we are setting up this in an industry, we don't have to worry about which energy source to provide them, if they are chemo energy source based, and if you have to give a certain chemical as an energy source. that we don't have to do, because they can just take up the sun's light energy to perform photosynthesis. And the other nutrients and electron donors and acceptors can come from the content of the flue gas itself, or even from wastewater, from different sources such as municipal wastewater, agricultural, and even industrial wastewater. And microalgae can convert this into biofuels, and even some of the cosmetic products, supplements, and in the end, the biomass can also be used as animal feed, because this CO2 from here is repurposed to other beneficial products. Another way of converting these complex emerging pollutants is methanogenesis. And methanogenesis is not a single step process. So many other things have to happen before methane can be produced. And methane is a common gas that escapes from many of the natural processes. Even though this is still harmful at higher concentration, at least it's not as harmful as some of the complex biopolymers that come out of the industrial processes. So initially, the first phase is hydrolysis, where the hydrogen bonds are broken, and they are broken down into monomers and oligomers. So proteins are converted to amino acids, polysaccharides into sugars, and fats and oils into smaller fatty acid molecules. So that hydrogen bond breakage is called as the hydrolysis process, and this can be performed by a fermentative bacteria. And another fermentative bacteria which can also convert these monomers and oligomers to even smaller compounds, smaller organic compounds such as butyrate and propionate. This process is called as acidogenesis. So these propionic acid and butyric acid are formed from these sugars and amino acids, again by another fermentative bacteria. And fermentative bacteria, some fermentative bacteria don't have to go through this in between process. They can directly go to acetogenesis instead of acidogenesis, where they can directly convert it into acetate, or convert it directly into carbon dioxide and water. So many bacteria can utilize sugars and process it and convert it into carbon dioxide and hydrogen. And acidogens are another class of microorganisms which are H2 producing, which can use up this acetate and also produce carbon dioxide and hydrogen. So these acetogens are relied on for this process. But this end step, where we want to convert it into methane, which can be used as biogas or natural gas as well, these are methanogens, which again are classified into acetoclastic and CO2 reducing methanogens. The methanogens can either act on carbon dioxide and then convert it into CH4, or directly act on acetate and convert it into CH4. So these are the two different types of methanogens that can be employed for converting carbon dioxide or fixing carbon dioxide to CH4, or fixing another carbon molecule such as acetate to CH4. But as you can see, different bacteria that utilizes different substrates and processes it into different products are required all along the way to fix this carbon dioxide that comes from these complex biopolymer products and convert it into something like methane. So this is how methanogenesis happens inside the body of the organism. These are all the different intermediate products that are formed from acetate or from carbon dioxide, and so many different enzymes are involved in each of these processes. I think we have gone over this various times now, that all of these are multi-enzyme processes. It's not just one enzyme that directly acts on the substrate and converts it. It's multiple enzymes that are involved. It's multiple pathways that are involved, and sometimes even multiple microorganisms are required to convert from one substrate to the final end product that is not harmful. So this is something we have already seen. So this just summarizes how various biomass can be processed by different processing bacteria, such as fermentative bacteria, transesterification bacteria, anaerobic bacteria, and biomass such as starch crops, agricultural waste, oil waste, so on and so forth, can be converted to bioethanol, biodiesel, biogas, and so on. All of this have applications in the energetics field, and hence any of these waste products can be converted to these useful products with the help of microorganisms. Bioelectricity production is also another application of how this carbon dioxide fixation can be useful for producing some useful byproduct. Here, acidophyllum is a species that is this is the cell of acidophyllum, and this whole process of conversion to bioelectricity happens inside the cell of acidophilium species, and it can be used for producing bioelectricity with a very similar process that we have discussed earlier, how the substrate is taken in and converted to bioelectricity. So the next few slides are going to be case studies of bioremediation. In the past few sessions, we have extensively looked at what are all the different bioremediation techniques, what are all the types of bioremediation, and so on. So we will look at one example of different bioremediation types, and where people have actually used this for real life applications. So the application of iron biocare composite in topsoil for simultaneous remediation of chromium contaminated soil and groundwater. So this was done in a particular place where chromium has been contaminating the groundwater, and this was done to immobilize chromium and make sure it is stable for a long time. And this process, it happens through reduction. So reduction is one of the strategies that we have seen that several microorganisms adopt to oxidoreductases perform this reduction reaction. And we have also looked at extensively about how chromium is harmful, how having chromium in the sediments is harmful to humans and the ecosystem. We have also looked at what are all the different regions in the country that are really affected by chromium in the groundwater and so on. So here in this particular case, they have done various processes including adsorption, reduction, and finally precipitation, even hydraulic flushing and leaching. So bioleaching is one of the techniques that we saw in the previous session. Maybe after I finish this, I'll probably go back to that and quickly go over that as well. So these are the different processes that is involved. So all of what we saw previously, everything is combined together and given here, so that we get an overview of how these different processes can be employed. Chromium immobilization due to reduction of chromium 6 to chromium 3, followed by co-precipitation with Fe3. So this is the process that they follow here. While chromium 6 is not soluble in water, it sediments and causes contamination for the groundwater. Fe3 is soluble in water and can be eliminated. So therefore chromium 6 is either directly or indirectly reduced and co-precipitated along with Fe3 um, ferric. So that's one of the examples of how they have a real life application of chromium contamination and how they have controlled it using reduction. So the next thing is also something to do with chromium immobilization. This is something that was done in a site called Hanford, and here they have used a microbe which can um, uh, use a slow release electron donor and then control the chromium. So this is Hanford is a region where a nuclear power plant is situated, and a nuclear reactor waste also contains chromium, and it mixes up with the groundwater in the lake in this site. So here they have followed the biostimulation process. So biostimulation is a process where there is an indigenous microorganism that is already available, and then there is the contaminant. So they add certain nutrients so that it can stimulate the microorganisms to start acting on the substrates itself. So here for biostimulation, they have added a slow release compound called polylactate compound, it's HRC, and they have used this to stimulate the indigenous microbial populations that are there in this water body. So this region is situated in Washington, US, and they have a lake where the Hanford is situated. So you can see here in this picture, this blue color is what the lake is, and this is where Hanford is, and they have lot of this waste from the Hanford nuclear power plant reactors here that get mixed into this. So they have added this polylactate compound, and this is what happens inside the microorganism cell, how chromium 6 gets converted to chromium 3, and how it accepts the electron the donors, and all of this process is explained here, and how chromium 6 reducing enzyme from the indigenous microorganism there is able to convert and co-precipitate Fe3, which is soluble. So the indigenous micropopulations are used to immobilize hexavalent chromium ions and convert them into chromium 3, which can be immobilized and can be attempted for long-term stable storage without contaminating the environment. So chromium 6 hexavalent chromium is the most harmful form, so they're converting it into chromium 3, which can later on be removed by other processes. So another example is an oil spill example. So oil spill is like a classic example that we have seen in the case of Pseudomonas fetida as well. So here as well they have used an oil spill, but this is a bioaugmentation process. So there was no indigenous microorganism to perform this. So especially in places like Alaska, which is a low temperature environment, extremophiles need to be added. Extremophiles are organisms which grow readily in extreme conditions, such as extreme cold conditions. Places such as Alaska are usually frozen; they're very cold. So selecting the microorganism that can process contaminants in that temperature is very important, and they have selected a microorganism which can perform this breakdown of the oil products in that region. So here as well they've used an indigenous microorganism, but the augmentation was done in case of providing the suitable nitrogen sources. So here the nitrogen source was the fertilizers that were added. So the fertilizers which contain or which are rich in nitrogen was added, and the oil spills in this region was contained using these additional products. Yeah, so that's mostly about the three major case studies that they have looked at, that we are looking at here for various applications of bioremediation. Maybe I'll just stop for some time for questions in case you guys have any questions before I switch to the previous sessions slides and just quickly go over that once. Are there any questions here in this? Because this is going to be our last session. Even if you have any questions from any of the previous sessions or any in general questions regarding the exams, please do feel free to ask them here. All right. I assume that there are no more questions in this session. So maybe I'll just share my screen and start with the session from yesterday, which many people missed. Because it's just to give a very quick overview for the previous session. So if my screen's still visible, just please do let me know. Can someone confirm?
Right, visible.
Yeah, okay, sure. So yeah, so yesterday's session was more about biomining or biohydrometallurgy, and also about some of the phosphorus and nitrate containing compounds and how to contain them, how to use microorganisms to remove them from wastewater and so on. So biomining or biohydrometallurgy is a process where we use microorganisms to extract metal from ore. So this is a continuation of what we saw in some of the previous sessions, where we have looked at how metals can be broken down by microorganisms. So biomining is one such process, and it is used in case of copper mining and many other metal mining cases. Bio leaching is different from biomining in the case that this is a special case where bacteria are used for utilizing metal sulfides as a source of energy and Fe3 as an electron donor, and how they can extract the metal from the metal sulfides. And here the example shown is a copper sulfide ore, and how bacteria can use this as a source of energy to convert it and extract the metal from this ore. So the difference between biomining and bioleaching is that biomining is like a blanket statement. It can be used for any metals to extract, and prokaryotes, any prokaryotes of fungi can be used for biomining. But bioleaching is a specific case where it is metal sulfides are used, and the organisms are usually bacteria that can perform bioleaching. And the bacteria are used for oxidation of the metal ore, and it allows us to extract the metal from the ore after oxidation, whereas in case of biomining, the prokaryotes take up these metal directly into their cell and then process it and then precipitate the metal after that. There are different types of bioleaching. The most common three types are the slope bioleaching, heap bioleaching, and in situ bioleaching. The slope bioleaching includes uh, uh, putting the metal ore across the slope, like a mountain or something that's closer to the area where it is found. Even in the picture we saw that it was like a slope where the mining site was. The heap is just, instead of a slope, the metal ore is placed like a heap. And in case of in situ, instead of digging out the ore and laying it as a slope or heap, it is done under in situ conditions, where the microorganism is injected into the site where the ore is present, and then later on, after conversion, it is recovered. The next thing is about phosphate removal process and nitrate removal. But before that, we saw what eutrophication is in some of the previous sessions as well. This is just because of enriched presence of nitrogen and phosphorus and other fertilizers. This causes the eutrophication, that is oxygen depletion, and algae and phytoplankton start growing in higher abundances. And eutrophication has serious implications on health of human beings. It has implications on the aquatic life as well. It causes increase in water temperature, toxin formation, foam and slime formation as well. So this can be controlled by removing the phosphorus and phosphates and nitrates, which are usually the contaminants in the fertilizers or whatever nutrients that gets mixed with the wastewater, and that is usually done by using microorganisms which can precipitate the phosphate as polyphosphate granules, and hence can be removed from the effluent before releasing it into the water bodies. So here, two different types of microorganisms are required. One is a phosphate accumulating microorganism, another one is a glycogen accumulating microorganism. The PHA that is produced in the anaerobic condition also has to be controlled, and the glycogen which is from the PHA also has to be accumulated and eliminated, and hence we require two different types of microorganisms. And the next is the biological nitrogen removal process. Another major contaminant is nitrates. And here in this case as well, we require two different types of bacteria. One is a nitrifying bacteria, and another one is a denitrifying bacteria. Here the ammonia is oxidized and converted to nitrate, and then the nitrate is converted to nitrate, and finally the denitrifying bacteria converts nitrates into N2 gas. Here enriching both of these microorganisms is important to convert ammonia successfully into nitrogen. So this is the process through which the ammonia gets converted to nitrogen, and this assimilation process happens through a bacteria known as anomox, which can convert ammonia to nitrate. And this reaction happens in a region called as anomoxisome lumen, or the anomoxisome membrane, which is the organelle where ammonia is processed by the anomox bacteria. So that was mostly what was covered in the previous session. So in case if you have any doubts from this session as well, please do ask now. There are no questions. So today will be the last session. There won't be any session next week. This will be the last session. And after this, I am hoping that all of you have registered for the exam and you'll take up the exam. So in case you have any doubts, please feel free to mail me or just mail the discussion forum. I will definitely get back if I have the solutions. Any questions, doubts? All right. If there are no questions or doubts, all the best. Thanks a lot for taking this course. And I know that the three of you have continuously attended all the 12 sessions. So thank you so much for that support as well, for attending every week session. I hope you found these sessions helpful. I hope I was able to provide some extra additional component along with whatever was provided in the course material. I hope these sessions helped a little bit to understand some of the concepts better. Hopefully, you'll continue taking more courses in NPTEL um, and learn a lot more from NPTEL courses. Thank you so much for attending these sessions. Am I still audible? Is everyone like there listening? Okay. Hello. I can't hear anyone, just in case if anyone's saying anything. All right, then I'm going to close the session. Thank you so much.